Thank you very much for giving me the opportunity to present today the PhD work that has recently been published, where we're looking at allosteric GPCR modulation in a retrospective, but also in a prospective way. So I think I don't need to convince you that GPCRs are a super important drug target. 36% of all FDA-approved drugs target them. And most of them actually, these ligands bind to the orthosteric site or primary binding site in purple here, and this is the dopamine receptor. And more structural information is out there that actually shows that there are alternative binding sites, allosteric binding sites, which are at a distinct location, but also have functional relevancy. And so these binding sites, they enhance drug response, but also open up new avenues for selectivity. And there are new also opportunities for chemical novelty and drug development. So personally, I've been very, very lucky to do now my PhD, because in the last 3 years, it has been marvelous. Every week coming out, new structures are coming out. And I'm not the first one in that field doing quite comprehensive analysis. Sorry for that. But we now have published a really most recent data set, which also Franco was using for his machine learning training. This data set actually highlights that these allosteric ligands, they're really binding over the whole transmembrane region. So for class A, class B1, and also for class C. And what is actually really interesting, not very complicated already, when you overlay these structures, you can see that there are certain sites that are really popular. And I really want to highlight a site, which is for class B1, this TM6 site. We heard it before, also in the presentation. TM6 swings out and the G protein can bind. And actually, that's the site that is super, super popular to be targeted allosterically, not only by negative allosteric modulator, but also by positive allosteric modulator, really stabilizing the conformation. And what is also really interesting is that we actually, for class C, we have functional dimers, and that we get also positive allosteric modulators in between dimer interface stabilizing these. We have a lot of knowledge, structural knowledge, yet the gap between FDA-approved drugs and what we are currently tapping is quite large. So I think that there's really big opportunity to dive into this space. And today I want to present to you a methodology that you can use to do comparative analysis of methods, and also prospectively ligandability assessment in structure-based drug design. And so this method is actually a GPCR binding site annotation scheme. This annotation scheme, it consists out of the class, the transmembrane helices that make up the allosteric pocket, and if it's membrane exposed, also the sublocation. This annotation scheme, it allows to systematically and precisely analyze allosteric binding sites and compare them across different studies. So nowadays, actually in our field, many people are using different pocket A, pocket 1, and this is really an effort to make communication on allosteric sites easier. And we at Nxera also implement this language already cross-departmental with medicinal chemists, also pharmacology and structural. And actually, it's really exciting. We're also collaborating now with the group of David Gloriam to actually introduce this binding site annotation scheme into GPCR-DB. So hopefully in 2026, with the new release, it should be out as well. So now let's go and dive deep into retrospective analysis. And even though we have this kind of exponential increase, we can actually see that there are these 100 structures that really bind to 24 sites. And these allosteric ligands, they cover intrahelical, extrahelical, and intracellular sites. And extrahelical sites, they are definitely the most diverse. So we were interested in looking at how kind of common binding site detection tools were actually performing, keeping in mind that these were actually trained often on orthosteric and not allosteric sites. And it was really nice actually to see that for most of them, actually, they're doing a pretty good job. But via GPS can detect 22 out of these 24 sites. And also what was really nice is actually that this benchmark data set that we now develop, we used it now to also improve FDMAP. And so there should be coming out soon a publication on that as well. This is just to highlight here that, yeah, we didn't detect 100 pockets. So yeah, it's a reasonable amount of average pockets for these binding site detection tools. So the next step was then, we have our allosteric data set. How are we doing on the reference data set? Can we actually predict these binding sites as well, if there is only the orthosteric ligand or the APO form? And so what we saw is that roughly 60% of the intermediate and inactive and 56 of the active allosteric binding sites, they are detectable in the reference. But one of the most challenging things actually that we saw is for extrahelical sites in the active conformation, and just added here 2 examples which highlight these induced fit effects. Another application of the binding site annotation is actually looking at properties of pockets, and that's especially important thinking about virtual library design as well. And so we see that these extrahelical sites, they're really in a chemically distinct space, very shallow, hydrophobic. And what was really interesting to us was actually that you get an enrichment for halogens. And so, yeah, especially with this kind of analysis, going back to the virtual screening libraries can be very, very helpful using very simply this binding site annotation scheme. So I showed you kind of more retrospective analysis. So let's move into the prospective. And for that, we use the binding site annotation scheme to look what kind of bioactive compounds, meaning having at least an activity at 10 micromolar from chemical, pharmacology and reaxis, can we gather, and that they are clustering with kind of known experimental structure sites. And we can see that already from that, we can actually form binding site hypotheses for ligands that are in the database that don't have at the moment a structure. So here, for example, for the adenosine A1 receptor, we get multiple other GPCR families that have very similar ligands, where we can then get an initial hypothesis of where these ligands could bind, probably at the allosteric site as well. Also for the intracellular CCR7, they're very similar ligands at CXCR1, CXCR2. And for the remainder of my talk, I want to focus now on cannabinoid receptor. And there we have a negative allosteric modulator binding between TM1, 2, and 4, so extrahelically. And what we see from the ligand analysis already, that there are very, very similar ligands also for the cannabinoid receptor 2, where there is at the moment no structural data. And looking at pocket, we can actually find also that BioGPS predicts a pocket at that particular site, really showing that that's very probable that this ligand would be binding there. On top of that, you get pockets for other GPCR families as well at the same location. And so we were then computing a 3D similarity score based on molecular interaction fields, and then also just a simple pocket residue sequence similarity. And what was interesting to see was beyond the cannabinoid receptor, we actually get the olfactory receptor and S1PR1 that have high 3D similarity, but actually quite low pocket residue sequence similarity. So that actually highlights that binding site similarity does not necessarily correlate with sequence similarity. And I think Franco's talk also showed that looking at protein language models on sequence level is not enough if we're thinking about allosteric. And if we look also at the sites per se, we see that it's actually key small residues. So in this case, glycine, which actually open up and really give the shape to the allosteric ligand. So in this case, S1PR1 would probably be an interesting target for further ligand repurposing, whereas for the other receptor, we actually get much more sterically bigger residue, and so that would be probably down prioritized. So with that, I hope that I could convince you that the binding site adaptation scheme has applications that are beyond the current knowledge. So it's actually hopefully applied to many more allosteric ligands that will be published, and that it really helps to benchmark tools that are out there, and also that these ligands, they're not binding there out of serendipity, but really are there for structural stabilization of key features for GPCR inactivation and activation. And yeah, that you have multiple orthogonal techniques that give you kind of a hint on the ligandability assessment. So with that, I want to just link you. So if you don't want to wait until GPCRDB has these site annotation, we published the code. So yeah, just go on GitHub and it just needs basically a GPCRDB for getting the generic residue numbers. And yeah, if you need any help, let me know. And with that, I would also like to thank you very, very much. Ejen, who's actually here as well from Nxera, also Giovanni, who's here from University of Urbino, and all of you for being so kind and listening to me. Thank you very much.
Next up, so there's time for a few questions. So, yeah.
Thank you. That was very nice. How many of these sites do you think you might have overlapping with cholesterol binding?
Yeah, for the cannabinoid receptor, actually, that negative allosteric modulator, there we know that there's also a cholesterol binding site. Yeah, I would say that there is, but it's a case-by-case study. You really need to take into account also kind of the crystallization conditions as well. So it needs to be taken into account. But I think, yeah, using orthogonal techniques really can help you to kind of choose that out.
Question from Patrick. Sonja, very nice talk. I'm wondering if it's, so from the drug perspective, I believe that it's very important that you find selective sites for Allostery, right? What you make with the annotation is that we have maybe found, you annotate them maybe more similar sites in GPCR, right?
So what is your question?
The question is that it makes for me, from a drug perspective, not so much sense, because what we need is, in my opinion, when we have allosteric modulators, maybe we need specific modulators for a specific target.
So I'm just annotating the location. I'm not annotating the properties.
But you know what I mean, yeah? So when I use an allosteric modulator and it binds to dopamine 1 and dopamine 4, whatever, yeah? Or it's a very similar site, then maybe this is not a help, in my opinion, right?
I think we're misunderstanding each other here, because the binding site annotation scheme, it's not like, so let's say we have a pocket at the extrahelical site where abacopam is binding. Abacopam is an FDA-approved drug. It's highly selective. It won't bind actually to any other GPCR class A, because there is a tryptophan that actually interacts via hydrogen bonding. So it doesn't mean that if we have a pocket at this site similar to abacopam, that our ligand that we are actually then designing for that pocket will exactly look the same. So I think it's more in general that probably the properties of the ligand, let's say hydrophobicity or halogens, that's something that you probably will need to take into account, since it's just simply a membrane protein interface. But I think that there's enough selectivity to actually then optimized for that. Yeah.
There was one question.
I think we have to separate location of the binding site from the nature of the binding site. Just because all ligands bind to extrahelical or TM345 doesn't mean that the property around that sites are exactly the same. We actually found that the location, the frequency of the binding site tend to correlate with the functional importance of the receptor. So that may partly explain why they are still druggable in terms of selectivity, because the nature, it's because it's very receptor dependent.
There was a question from Jaya.
First of all, thank you very much for this very interesting presentation. I have a question for you and the previous speaker, which deal with an issue where I'm not a specialist, but which I found was debated in the past years, which is the stoichiometry between the G protein and the receptor subunit. And in all the scheme you present, even when there is a dimer, you show one molecule of G protein. And apparently the stoichiometry is not 1-to-1, but 3-to-1, like in the case of fluorescein in the retina, or other kinds of stoichiometry. So could you comment on this issue, and in particular, the fact that you, for the dimer, you use the same stoichiometry. And normally there should be two G protein associated with the dimer, according to the 1-to-1 relationship we see to be controversial. So, what is your opinion about this issue, and also the fact that in class 1, like the beta-adrenergic receptor, it has been debated that the formation of the dimer is transient and difficult to capture. Maybe this is wrong, I don't know. So what is your opinion?
So Sonja first.
Yeah, so I really looked at the static structures and kind of just based my analysis on experimental experimental structures. So there, obviously, your question is highly dynamical, and I think the first speaker would probably be better at addressing your question. Thank you.
Your comment? It's about the stoichiometry between the receptor and the G protein. So,
What about the previous speaker?
yes. Repeat the question briefly. Repeat the question, please.
So the question, I repeat the question, is there a same stoichiometry for the G protein between the monomer and the dimer? That's the thing. And furthermore, from what I learned in the past from colleagues, there seems to be not a 1-to-1 stoichiometry, but 1-to-3 or some other type of stoichiometry taking place even for the so-called monomer. And also the so-called monomer could, in fact, in the case of class 1, be transient and not see, except perhaps by your technique. I don't know. So, what is your opinion about the stoichiometry of G protein to receptor, and in particular, the case of the dimers, which are well established in class 3?
So, as you correctly pointed out, in classic GPCRs is the only de facto dimer. This is because they're disulfide linked. There are cases, very, very few. I only know of chemokine receptors where they have shown that there is some regulation between protomer, dimer, and trimer. I would argue polymerization in many other GPCRs has been fiction, and a fiction that has been propagated from the early 2000s. The minimal functional unit is a single transmembrane protein coupled to one transmembrane coupled to one G protein, and we have seen it working. We can reconstitute it, and it is working as it should. I am not saying that proximity from other, the same or even other domains from other receptors can influence function. But in terms of having a dimer and a trimer, it has not really been observed other than in chemokine receptors in the de facto family.
Yes, but the criticism we should make is that you are looking at reconstituted system and not physiological one.
You're absolutely right. But these observations are corroborated by single molecule threat measurements as well. And again, you're going to see in that theory, sort of in that line of work, aspects where people say, oh, it could be oligomers. Sure, if you're going to overexpress and do single molecule threat on the plasma membrane, you have packed so many receptors that you don't have the ability anymore to separate monomers from dimers.
Thank you very much.
It is a pity that Julia Ossetti, part of the network, is not around today, so she will cut more. It is around because she's working on the dimerization of GPCRs and how dopaminergic receptor